Hi, Desmond. How are you doing?
I'm good. How are you?
Good, good.
I have,
Very excited to be here.
yes, I have two Zoom sessions going on at once.
Oh, wow.
Yeah, I have two students that are taking an exam. So I think, guys, it looks like it's working. So we should be good.
It's
And they
working
might
on
be
our end.
finishing soon. But anyway, all right, so today it's kind of interesting to me that I think was 2 weeks ago now, we had a seminar on nanoparticles, but that seminar focused on toxicity of nanoparticles and following up, making sure that they are safe in us using them in research and in commodity items. Today, um, is an application. So that's kind of interesting in that back to back. Our speaker is going to be introduced. Our speaker is going to be introduced by Dara, and Dara is going to actually tell me her pronunciation of her name for maybe the 100th time correctly. But Dara hails from Jakarta, Indonesia. That's a place I'd like to go actually and check it out. She's a senior biochemistry major. Her high school was Bina Bangsa School in Indonesia. And actually, one of the really cool things that I know about Dara is we have this public science thing that we do for every student that does research with me. They have to do like a video, a research video, a blog. Dara decided to do an artwork. It was cool. It is cool. So she has that talent, and that's one of her hobbies. Um, she plans to go to dental school after leaving Andrews, and hopefully to specialize in orthodontics. Dara, it's your turn to introduce our speaker.
Thank you, Dr. Murray. Um, so my name is pronounced as Dalantea Atmaja, and I'm going to introduce our speaker now. So Dr. Natalia Gonzalez-Pech is originally from Coatzacoalcos, Veracruz, Mexico. She received her bachelor's in chemistry from Monterrey Tech in 2016, and she completed her PhD in chemistry at Rice University. Her doctoral research focused on nanomaterial synthesis and characterization, and their applications in water processes. Dr. Gonzalez-Pech did her postdoctoral research at UCSD, which focused on characterizing metal-containing nanoparticles formed in industrial processes as well as the understanding of their health effects. Currently, her research group focuses on the development of nanomaterials for water remediation and energy-related applications in a sustainable manner.
Thank you for that introduction, Dara. I'm really happy to be here. I'm going to be talking a little bit of what we are doing in my group at Hub College. I currently have 6 students. They're freshmen and sophomore and junior. So they have been with me for a year, and I think we have accomplished a lot. But in order to me to talk about what I have been doing or we have been doing in the last year, I want you to give you the motivation behind. And the motivation behind started actually basically 10 years ago with my work as a PhD student, where I focus my research on development of iron oxide nanomaterials for technology for arsenic remediation. Why arsenic? Well, as you maybe most of you know, we have a lot of contaminant in waters. When we think about drinking water, most of the time we are looking at microbes because it's what makes us sick very fast, right? but also we have disinfectants and disinfection products in there, and also inorganics that sometimes we don't look for them because we find them in very low concentrations, and that is the case of arsenic. The issue with arsenic is that if you are drinking it at very low concentrations, it damages every single organ in your body, and it have a horrible death rate for cancer. And people realize only that they have been poisoned by arsenic when they start to have these horrible dermal effects. Now, that is the way that they found that a lot of thousands of people in Indonesia and India were being poisoned with arsenic because of they were drinking it. But that is not the only case. We can find issues with arsenic in Asia, in Africa, in Latin America, even here in USA, in Arizona, New Mexico is a big issue. But it's not as important because we have other resources for drinking water. Arsenic in water is a kind of complex system with depending of the pH and the redox potential of the water. We can have different species. Most of the time, this red area is the one that we can find in groundwater. So that means that arsenic can be as in its state 5 or 3 as oxygen ions, and it can be the monoprotonated or deprotonated. So this makes the problem complex, right? Because instead of thinking about one single thing that you have to remove, you have different options and the interactions are different. We can complicate this even more when we look at the conditions where we find arsenic in Latin America. Most of the papers right now focus on solving arsenic in Indonesia and India because those are where a lot of people have suffered, but those conditions are reducing. So, and we don't have many interference. In Latin America, we have very oxidative conditions that if you see is because every single place that we have found arsenic is very close to volcanic activity. So that means that we will have a lot of silica that kills any material that you want to use for arsenic remediation. You have high salinities, high temperature, so the problem becomes even more difficult. And when I started working on this project, we have a collaboration going on in Guanajuato, Mexico, where we were able to go to Salamanca City, and the concentrations in all the wells that we sampled were between 25 and 100 ppb. To give you an idea, the maximum concentration level that USA allows in water for arsenic is 10 ppb. So every time that I was in there making some analysis, I knew that the coffee I was drinking had a lot of arsenic. There is not a single solution for arsenic remediation. We have a lot of options, but depending of the conditions that you are trying to solve or the different steps of each of the techniques, or if we consider the implementations, do we want to use them in a large plant or a small well, the interfering species, and how much money we want to spend, we decide that our absorption is probably our best shot. The cheapest one, the more efficient, and we can play with the materials. And when I joined the lab, they were synthesizing already iron oxide nanoparticles, and they have this nanomaterial that they knew that they could use for arsenic. So that is the way that they approach the problem. They found that they have really good performance, right? But that was all. When you need to bring something to the field and solution that you think is a possibility, you need to think about the problem in a different way. And this is what a lot of people call design for purpose, where you need to define a problem, design the material that can solve that, go and do it in the lab, and then you will find that you have reality check, like how efficient is my synthesis, how expensive it is. And then you go and check if the material that you synthesize is a product. Of course, we never go back in a single, we never find the right solution in a single assessment. We have to go back and redesign, right? And depending on the reality checks that we find from the synthesis, or then if we have that bad performance, then we have to go back and redesign things. And that is the way that I look at my work while looking at synthesizing materials for arsenic remediation. So to define the problem, we are looking to remove arsenic from groundwater. We know that in those conditions, we have arsenic 5, we have high pH, high temperature, we have a lot of silica, also vanadate around, and we want to use particles, right? Nanoparticles, you knew that those are good because we have a lot of surface area where they can absorb a lot of arsenic. We want to use iron oxides because those are materials that have this structure on the surface. We have the iron hydroxylated, so it's neutral. And at those, the pH that we find in groundwater, we can generate this bond with arsenic that is very strong. The only way to break this bond is by a lot of very basic conditions. So once that we are able to absorb it, it won't be leaching. So that is a good thing. During the synthesis, I did several kind of synthetic methods: thermal decomposition, coprecipitation, and sorbothermal. I'll be talking most of the time about sorbothermal later on, and everything, all the synthesis was characterized by TM, SEM, XRD, Raman, XPS, IR, and BET. The reality checks that I focused on were the cost of the synthesis, the technology implementation. What do I mean with that? Can I really bring this nanomaterial to the field and solve it and solve the problem? That is something that as a field we are still dealing with, and that is one of the things that I'm trying to change with my research program. And the other thing is how much it costs me to treat 1 liter of water, right? Finally, for performance, when we are looking at absorption, we need to do absorption isotherms. That is a batch experiment that look like this. Then if these results look good, we do the small columns that I can do in the lab. If this goes well, then I can do pilot scale columns. And if this goes well, then I can implement in the field in a inline well filtration. Because I'm a chemist, my work focus on these first two. And the way that I do the absorption isotherms is you weigh the absorbent, you expose it with a solution that has arsenic, you remove the solid, and then you measure the amount of arsenic that you have. Based on that experimental data, you try to feed it to a Ladmure isotherm, where two parameters are important: the maximum capacity that the material can absorb. In this case, for example, a bigger capacity gives me this black line instead of the orange, and a larger Ladmure constant that is basically how fast I can saturate the material at lower concentrations. The other kind of experiments was the column experiment. When we pack our material in the column, we flow water with some pumps in there, and then we collect the solution after it passes the filter. We make sure that we don't have any particles in there, and we analyze the concentration of arsenic. So what we plot is basically how much time or how much volume we can treat, and how much arsenic is getting now over the amount that I put in at the beginning. A typical experiment, a typical column should look something like this. And this curve is given by the advection dispersion theory. And two factors are important in here: the retardation factor, that is basically how much time I can treat everything without starting to see arsenic increasing or any analyte increasing. And the other one is how the diffusion that is telling me how fast this from will come, how fast I'll saturate. And this, we feed everything to this solution. I'm not gonna talk about the mathematics, but the idea is that, well, something that looks like the green is good, something that looks like the blue line is better. We want not to see increasing in a concentration. Something that looks like the red line is horrible, right? We are not really treating any volume in there. So the way that I did the projects, when you're doing a PhD, you do several things. First, I wanted to evaluate what was the problem. And with that, I used arsenic sorbents that we can find in the market. I bring them to the field and see, okay, how much arsenic they can absorb in those conditions. In there, we found the issue that silica was and how it was a big interference. Then, but I'm not gonna be talking about that in detail. Then the next thing that I wanted to know, okay, can we put the nanoparticles in a column? Can we really use them in real life? And in here, I focus on aggregation of nanoparticles and how to prepare the column. And I compare 3 different kinds of synthesis. The thermal decomposition, where you can see that you get very nice nanoparticles. They are fully dispersed. This is something that you prepare in a hot reaction in organic. Then co-precipitation, you still have really nice particles. The issue is that they tend to agglomerate in these big chunks. And then the hydrothermal, or the commercial one, this one is a material that you can go and buy it. And you can see the nanoparticles are around 100 nanometers, but they are very, very agglomerated. So the first thing I want to do is do arsenic absorption, isotherms, and we can see that the nice materials have the better performance. We have a big isotherm here, followed by co-precipitation and then hydrothermal. We do the same with silica present, and we can see that, yes, in these conditions, we have a slightly decrease in absorption capacities, but it shouldn't be as big, right? The main factor that we have to worry is about agglomeration. Then we try to prepare columns. This is a commercial material. This is what we want the column to look like. We have some support, but this brown thing is or the material, and then we want to pack it in the column. For that, to generate this amount of nanomaterials is a lot of time doing synthesis. But for example, the thermal decomposition are nanoparticles that we generate in a solution. So what I thought at that moment is like, well, we have these nice beads, polymer beads that we can use as a support. If we start the nanoparticles on the surface, I start to saturate this one. And as much as I add, I think the highest that I added in here was 2%. And we can see how the bead changes before adding the nanoparticles to adding them. Now we have a lot of contrast coming from the nanoparticles. When I try to do columns in here, as you can see, the co-precipitation and hydrothermal, the column experiments look horrible. The arsenic comes very fast. But when I do the thermal decomposition, I cannot even flow water. Why? Because the thermal decomposition of the nanoparticles, you have a surface coating that is organic. So I was created a very hydrophobic material. So the next step is like, okay, I'm gonna remove the coating and mix it with the beads. And when I do that, you get exactly the bad behaviors that you see of the other materials that are agglomerated. So one of the ideas that I have at that moment is like, okay, this is a polymer. I can remove the coating from the nanoparticles once they are in the beads. And the idea was to try to image them. Those white spots are the nanoparticles, and it's bright because of the organic content. When I reduce that coating, we can see the particles depositing there, but this treatment destroyed the bit. So in the moment that you want to run something, it's basically a gel in there that doesn't let water flow. So because of that, we moved to sand, where I can do that kind of treatment without damaging the support. Instead of going to a 20%, because this takes a lot of material and time, I did a 1%. And if we look in the tail, we can see that it's not a good column. Well, maybe co-precipitation is, but I wanted to look how this look in the sand, because the beads are very spherical, very smooth, very homogeneous. Sand is so smooth, but still you have different areas, right? And these roughness that you see are coming from the nanoparticles. You cannot really see the nanoparticles in here, but you can see how the contrast changes. So what we hypothesize is that you have certain amount on the surface. In the co-precipitation, you can see the agglomerates of nanoparticles. These are bigger in the hydrothermal. If we go and quantify how much arsenic we are absorbing, even when the columns are not that good, we can find the thermal decomposition, the ones that we have nice nanoparticles and they are deposited in an homogeneous well, can absorb much more than the co-precipitation that was giving me something. So then it's like, okay, we need to find a way to use the nanomaterial without generating a huge agglomeration, right? And that is the first redesign that I have to go back and do. For that, at that moment, we found a different synthetic methodology to create clusters of nanoparticles. And I work on not only assessing the arsenic removal performance, but also to understand what was the mechanism of that synthesis. And when we talk about nanoparticles, if we are using a synthetic method that is inorganic and very fancy, we will get something that looks like this, right? You have the nanoparticle that is well-defined, and you have a surface coating that allows the nanoparticle to be by itself. If you remove this coating, then you start to have a small agglomerates. Maybe 2 or 3 nanoparticles, but still, you cannot find them individually now. The cluster of nanoparticles, what we did with this is, let's use that agglomeration. Let's create a material where we are defining that agglomeration, that we can control how big the agglomerate is and that is well defined. And we can combine and we can change the parameters and may have clusters that are as big as 100 nanometers and nanoparticles, individual nanoparticles that are like 5 nanometers, or clusters that are 100 nanometers but are made of nanoparticles that are 20. Or even I can do very porous materials with this synthesis. So I play a lot with this synthesis. I won't go through all the parameters that I move, but you can see that, for example, by changing the amount of polymer, I can break a little bit this interface, and I can see that my material is hollow. I can also, one of the studies that I did is follow the synthesis through time, so I can see how the clusters are being formed, and then how with more time we get a more homogeneous distribution. We also look at different temperatures. After it's 180 Celsius, we can start seeing magnetite instead of iron oxide, iron 3 oxides. We can control with changing temperature. We can control the size of the cluster and the nanoparticles. We can see the conversion from the iron oxide 3 to iron oxide 2, 3. And based on that, we propose a mechanism. We have an amorphous mixture that we are heating a lot, and we are putting in a high pressure in this vessel, and those bubbles are crystallizing slowly from the surface, and the material is the amorphous material is growing these crystals. When I went and see the absorption, the performance, the arsenic performance, they were a little bit disappointed. I was trying to do these studies at the end of my PhD, so I didn't really have time to optimize this and try to understand why this was happening. But one of the things that I wanted to make sure is like, do having the clusters of nanoparticles allow me to run columns? Using this, I didn't have any more issues with water pressure. That is what happened when you have nanomaterials. But still, the performance in columns was bad. But I found that even when the concentration increased very fast, it never fully saturated. We have this area that, looking at the literature, looks like this is a ionic exchange behavior. And I want to look at the IR and see, okay, I'm using a polymer as a precursor. Of course, I still have it in the surface. So when I'm flowing arsenic, some arsenic is being absorbed. That is what is getting in here. And we are releasing that polymer. That's the reason we have this behavior. And I went and confirmed that by analyzing how the vanadate was behaving, and it's the same. We are not saturating. So that makes sense. So that was a thing that I needed to improve. But again, I was trying to finish a PhD, but something to consider. Another thing that I tried to do during my PhD was decrease the price of these nanomaterials. At that moment, the group have a kitchen synthesis that was amazing. You can do nanomaterials with olive oil and things that you can find in your house, but those nanoparticles didn't absorb anything with arsenic. After the modification I did, we got really good performance, but because of time, I'm not gonna talk about that today. So that was my PhD. So I was able to generate nanomaterials. We were able to understand how the parameters of agglomeration influence in the arsenic absorption. Still, I have that feeling that, okay, I tried to bring some nanomaterials to the field. It didn't work. I kind of understand why. And now that I'm with my group, I'm going back to there. But before that, I went to San Diego to do my postdoc. And what I decided to do with my postdoc is move from application to implications. I was using before nanoparticles to improve the environment, right? Try to solve an environmental problem. But nanoparticles can also be a little bit bad if we are not careful of how we handle them. So that is what I focused during my postdoc. We know that now with the nanotechnology being so advanced, we are being exposed to more nanoparticles. Most of these nanoparticles we engineer, and we can control that exposure. For example, some medicines are using now silver and gold nanoparticles, and you will go and inject that. So you should be able to expect where they end up. But what happened with the nanoparticles that we are exposed by accident? Like, for example, like if you are doing laundry and now they are these antibacterial fabrics with silver nanoparticles, and you put that in the skin. It's like, that is not something that people is not considering, right? Even worse, what happens when you deal with incidental nanoparticles? Incidental nanoparticles are nanoparticles that are being generated in process without us really knowing. For example, in any combustion, we can find fly ash. Those are carbon nanoparticles. Industry generates a lot of metal nanoparticles, mining. Even your car generates nanoparticles. And we are breathing those airborne nanoparticles in the same way that right now we are worrying about coronavirus. It's the same size. We are breathing that in the environment. And the issue of that is that these nanoparticles can go to your nose and move through the olfactory bulb to the brain, or you can even breathe them and go to the deep of your lungs and translocate from there. So a lot of people know that. Several people have studied transport of nanomaterials of particles in pollution. We know that we have a particle matter 10 and particle matter 2.5, that what you are doing in there is defining how big in micrometers they are. But lately we know that you can also find nanoparticles in pollution. And when I was starting my postdoc, this study came to light where they found magnetite nanoparticles in human brain. They were doing autopsies of people that died in Mexico and in UK, and they found nanoparticles, and they determined that they weren't biogenic, means it wasn't generated by your body, but it was something that was coming from pollution. And that made me think, okay, that is iron nanoparticles that you find in pollution. What happens when you are getting exposed to nanoparticles? For example, if your job is generate titanium dioxide nanoparticles, and there is an accident and you end up breathing a lot of them. Or one of the things that worry us was manganese oxides. Manganese oxides are one of the nanoparticles that you generate while welding. And they did a study about how welders, they actually see a change in the magnetic response of their brains. Imagine how worry is that. So I decided when I started my postdoc to have these 4 questions. I was worried about what nanoparticles we have to worry in occupational settings. Can we predict their behavior during the entry road when we are breathing them? And do we understand interactions that move them and therefore the health effects? So for the first questions, what I did, well, my collaborators did, were to different test sites, a machine assembly center, an indoor shooting range, and an iron foundry. And they started to measure how many nanoparticles they get through different days. And as you can see in here, there are a lot of them all during the day. They were measured by a scanning mode for me, where they pump the air of the environment and deposit them. Oh, okay, I'm back. And deposit them on these filters where we were separating them by size. And once I digest this, I can go and look at the composition. And right now I'm showing the ones that are in higher concentration, but we analyzed more than 30 elements to make sure that we were finding everything. And looking at this data, it took me a while, but one of the things that came to mind is like, why are we seeing similar distribution? Does this mean that we have a nanoparticle that have both iron and manganese and copper? Or we have nanoparticles that are iron and others that are manganese and others that are copper? So that is something that we wanted to find out. So we went and do microscopy in the different levels. And for example, this one that is around 5,000 micrometers, they should be big particles. And we found them, but when we look closer, these have these nanoparticles on the surface. These nanoparticles looks like the nanoparticles that we were seeing in filters for a smaller size. And this is in the heavy vehicle machine assembly center. But the same happened when we go to the iron foundry. In the iron foundry, you have a lot of silicate, so all that amorphous thing that you see is a lot of powder, basically. But again, we have these big spheres that are iron, and we have other nanoparticles in there, and you can see even particles over the particles, right? And that it was like a little bit, okay, maybe this is something that we should have expected. You can have agglomeration in there. And it's like, okay, one of the things that we wanted to find, do we have different, are these nanoparticles different metals, or is all combined? Based on the size, well, for example, we have some small spheres and even smaller ones. We have some cubes in there. We decided that each of them are a different metal or metal oxide. And that is interesting. Now we know what we can do, but this kept me thinking, can we predict what happened with these particles when we breed them? Before, or there is this theory, or this has been studied for years now, that depending on the size of the particles is how far they will go in our lungs, right? If they are bigger than 10 micrometers, then we probably will inhale them, but our nose will filter them. If they are smaller than 10 micrometers, then they go in the thoracic part. If they are even smaller, they can go deep in the lungs. So depending where they end up, depending on their size, they end up in different areas. We will expect that the big particles that I saw end up in here, and probably they are cleaned by phagocytosis. The small nanoparticles can go deeper in the alveolus, and they can cross the cellular barrier. So they can come and be in the bloodstream. And that's the worry. So one of the things I wanted to do, okay, what happened with them, with those nanoparticles, if they are cleaned by phagocytosis or if they go to alveolar uptake. So what we did is studies of kind of mimicking the conditions that you will find in macrophage or in the interstitial fluid, that is basically the fluid in the cells. And for this, we can do solutions that are mimicking those conditions. One is the artificial lysosomal fluid that is at pH 4.5. We have a lot of organic in there simulating the protein binding that we will expect. And then the Gumball solution that is something around 7, also with a lot of organics. And we were trying to mimic to the nanoparticles or the particles that we found in the real life conditions. To that, this is how they look from the real welding. This is what we were able to generate in a spark discharge generation chamber. And we went and exposed them to those solutions, trying to mimic the physiological conditions, right? And we found that they only dissolve partially in the alveolar, in the ALF, that is the macrophage conditions. So that means that if the nanoparticles cross the barrier and go to blood, they will enter our bloodstream. Looking in the tail of these nanoparticles that we generated in this synthesis, we found out we have 2 different oxides in there for manganese. So I wanted to make sure and understand which one we were dissolving. And the manganese oxide, manganese 2,3 oxide, doesn't dissolve at all, while the manganese oxide 4 dissolves in these conditions. So that means that depending on what the nanoparticle, what is the chemical composition of the manganese in the particle, it will dissolve or not. And I also have other projects, and I look at interactions with proteins and amino acids. The idea was trying to predict where they will end up. I'm not gonna talk about that because it's not part of the story that I want to tell you. But in general, during my postdoc, I characterized nanoparticles in occupational settings, particles that workers are breathing. We found that they agglomerate in these conditions, and we try to correlate the surface chemistry of these to the implications for health. And all this was work that I did during my PhD and postdoc. I have two advisors, Vicky Colvin and Vicky Gracian, a lot of mentors and collaborators. I work very closely with a lot of grad students. But one of the things that I enjoyed the most was working with undergrads. As you can see, I worked with more than 10 during this, before I was a professor. So that's the reason I decided that I wanted to look for an institution that allowed me to keep doing research, but also interact with undergrads. And the research that I wanted to do was based on my background, where I know how to do nanomaterials. I do a lot of synthetic work. I want to synthesize materials that I can bring to the field, but I also have this, I'm very aware how these materials can end up affecting life in other conditions if we are not be careful. And also when looking at these materials, the nanoparticles that I found in air, this structure where we have multiple metal oxides worry me a little bit, because, okay, you are breathing something that is very reactive. When you have different particles in there, this interface can be very reactive, and you cannot really predict what is going to happen. So when designing the research that I wanted to do, this was an inspiration. Okay, can we create something that we have multiple interfaces for environmental applications? And that is what we are doing at HOPE. That is what the NanoGOSH group is doing. We are developing nanomaterials for water remediation or energy related in a sustainable manner. I want to make sure that every material that we are doing, we can bring to the field. And these are the first results from my group that I'm very excited about being able to share this. These are the first clusters that my students did. Very nice. And these are the latest one. In here, we are starting to incorporate nanoparticles as precursors. You can see how these clusters are very homogeneous. The nanoparticles are very small. That is what we did before, right? Now we have this cluster that is not as nice, but still you can see that you have an homogeneous distribution in the nanoparticles in there. And then you have these big particles, or this one, you have this one, the yellow arrows are pointing the ones that don't fit in there. Why? Because those are nanoparticles that we added. And the idea is that without the nanoparticles, we will be forming these. If we added, we can put the nanoparticles inside of the cluster. And we corroborate this by DLS. We added, this is our precursor nanoparticles. Then we go and generate the cluster, and the nanoparticles are not free anymore. And this is very exciting because now I can change the composition of these particles, and I'm generating different materials, and the possibilities for applications are widely open, right? Then I talk about how we did experiments with clusters of nanoparticles. This is a graph that I have shown you before. The one that we saw that we have issues with the surface of the clusters. This one I haven't shown you before. This is the first column, that experiment that I have during my PhD. This is a sorbent that is commercially available, but treating 1 liter of water will cost us $10. So that is not feasible, right? And this is the column that we just analyzed this week. Like, we are still not there, but it's the first experiment that we run. So we know that the materials that we are doing and removing arsenic. So with that, I want to thank you and thank my students. I think we are doing good progress, but we are also having fun and we are enjoying this process. And if you have any questions, I'll be more than happy to answer them.
Very good, very good. Thank you very much. So questions, anyone? I'll begin. So in terms of the types of metal oxides that you're working with, it seems like most of them are transition metal oxides.
Yes.
Is there any interest in your group or in the field for like non-transition metal oxides?
There's definitely interest. I look at transition metals, mainly iron, because it's cheap, right?
Yeah,
Iron is something that we can find a lot. So
right.
that is the first thing that you want to make sure that your nanomaterials are, if you want to use them in real life, that they are cheap. I like to work with transition metals too, for example, manganese. With all the oxidation states, you have a lot of possibilities in there. Even when they are not the best catalysts, but when you have these interfaces, I'm hoping that that interface will improve any performance in there a lot.
Okay.
But yeah, we could do the same kind of materials with any other no transition metal.
Okay. Okay. You guys could put questions in the chat or you could unmute your mic. Another question is about titanium oxide and zinc oxide, because those are fairly widespread in commercial applications, correct?
Sorry, my internet froze for a minute. What did you say?
The titanium oxide and zinc oxide, are they pretty prevalent in commodity type products?
Yeah, that is definitely something that we are already working on.
Okay.
We are very excited about looking at how the photocatalytic properties changes when we have the iron combination in there. And we are preparing big particles that we can use in filters in water. And I'm sure that we will be able to radiate them and see the composition of organics, for example.
Okay. But has there been any work in your group or other groups on the toxicity of those two? Because it's claimed, I think it's claimed that, for example, the zinc oxides, nanoparticles are fairly non-toxic. Is that the understanding in the field right now?
Yes, the good thing about zinc oxide is that it dissolves. So it's very reactive, right? You want to use it as an antibacterial because will go and interact with the cellular barrier and destroy that. But once that they interact or they are in the media that you want to use it, it will dissolve. So that's good. Titanium dioxide, a lot of people have studied them. And what happened with nanoparticles in the environment most of the time, they tend to agglomerate. So even if you use them in, for example, sunscreen,
right,
it will end up in the sea, but then it will deposit to the soil. While I was doing my postdoc, we were doing a studies of, well, what happened once it's in the sea, right? And we found that they might come out, but the theory so far is that they deposit and they are safe.
Okay.
Oh, yeah.
Anybody else wants to chime in?
So if these, in commercial use, how do you see these nanomaterials being recycled?
For example, in the case of arsenic, right? That is one of the questions that I always get. Like, okay, once that you remove the arsenic, what happened with the material?
Right.
To recycle, you have to pass a very basic solution, right? And that is the way that you clean the material.
Right.
But yes, it's not, at the end you will have a solution that is very concentrated, but then it's better to have 1 liter of something that is very concentrated, right? Versus having tons of liters that are being consumed.
Right.
The materials, one of the things with materials is the more you expose them to something, the more they absorb.
Right.
So right now I want to make sure that when they are exposed to 100 ppb of arsenic, they absorb as much as possible, right?
Right.
But if I use the same material that is spent to clean something that is 10 ppm, they will still absorb more. So at some point, we will end up with solid with arsenic, but it's better to have it concentrated and it's easier to manage.
Right. But
Yeah.
and that's great news about logical about the arsenic, but the nanoparticles themselves are permanent. I mean, they will never degrade. What's happening with them once they lose their efficiency, or is there anything like that?
Yeah, I think, I mean, to regenerate them, you have to make it harsh conditions, right? So
Right.
if you do that several times, I'm sure that we are gonna lose that nanostructure, and you will have iron oxides. But it's one of the reasons I like to work with iron, because it's something that is an oxide. It's something that you can find in the soil. So as
Sure.
long as you manage whatever you are removing correctly, it should be okay. We are not generating something new that we will worry about.
Okay.
Yeah.
Thanks.
So have you also been playing with mixed metal oxides?
So right now we're moving to mix to have this support of iron oxide. We are working with zinc oxide nanoparticles, titanium dioxide nanoparticles. We are at some point, once that we have the synthesis well controlled, I want to start adding not only one kind of nanoparticles, but what happened with 2 kinds, right? And how that plays. We are slowly progressing in that direction.
Okay. Yeah, because those systems could be potentially very good in terms of catalysis, right?
Yeah, that is the goal.
Mm-hmm.
Yeah.
Okay. Any other questions? All right. If not, I know one of the things that I was interested in, especially when you started talking about arsenic, we did have a project, environmental science type project in my grade 10 class. And one of the things my students there found out was the problem of arsenic in things like baby food.
Oh, wow.
Yeah, exactly. So yeah, that kind of perked my interest. Are you looking at some of those sort of applications in, I don't know how you would apply it to baby food, but baby food, from what we understood, came from these rice fields.
Okay, that makes sense.
Yeah,
Arsenic in rice is a big problem. It's something that
right,
people have been looking at in the field.
right.
The issue is because you are planting rice, right? It's coming from the water that the plants are absorbing from growing. You
Mm-hmm.
actually have the same problem with wine. Depending on where the wine was, the grape was cultivated, you might have arsenic in there. So yeah, be careful with the one that you are drinking. But yeah, I mean, definitely the way that you have to think about how to solve that problem is different than with water. Yeah, we will have to talk a little bit more about what we can do in there.
Okay. Yeah, it's a problem that affects, especially in the case of the baby food. I mean, we don't want to be
Yeah.
poisoning our babies. That's a bad way to start your life,
Yeah, no, because even arsenic, that is the issue with arsenic. High concentrations will kill you fast, but low concentrations will generate problems that can last decades. And,
Yep. Yep. All right. I think we will end here, but not before thanking you again for a beautiful, wonderful seminar. Very informative, very relevant to different aspects of chemistry and health. So thank you very much. And I did promise you that I'll be contacting you again to do a talk for my high school kids.
Sounds good.
All right.
Thank you
Thanks.
so much for the invitation. I really enjoyed joining you guys.
Okay. Have a wonderful evening.
Thank you.
Okay. Bye-bye.
Okay. Thank you for that,
All
Desmond.
right.
Let me know
We
when
will
you
we will be in touch for sure.
sounds good. See you.
Okay. Take care.